Bonsoir, donc bienvenue pour ce dernier cours d'Emmanuelle Passegué, professeur invité donc au Collège de France par l'Assemblée des professeurs, qui va nous parler donc aujourd'hui d'un sujet qui nous préoccupe beaucoup, le vieillissement et la réponse au stress. Et avant de te donner la parole, je vais d'abord te remettre la médaille du collège, voilà, qui est réservée à tous ceux qui sont invités pour faire un cours comme professeur invité. Merci d'avoir été avec nous. Et... Ah. Et donc, Emmanuelle, nous t'écoutons.
Merci Hugues. C'est un tel plaisir de donner ce cours et de finir cette série de lectures sur la question du vieillissement. And the title is Hematopoietic Stem Cell Aging and Adaptation to Stress. So we all are concerned, especially in the scientific world, with the same question. Basically, why do we age? And how can we live longer, healthier lives? So what can we learn from the biology to try to tackle this fundamental question? So why do we age? It's because mainly of medical progress and the ability of decreasing a lot of early lethality, infant death. So treating problem with infancy, development of increase, I'm sorry, I'm mumbling my speech, increase medical care, but therefore population globally is aging longer. And with increased aging, what we see is very much this increased rate of a lot of disease of old age. So in medical term, aging is an increase of a rate of risk of dying, which basically in a given year doubles approximately every 8 years. So the older you get, your risk of dying increase. At the level of the organ, what we see is, as stated in textbook, a progressive and complex process whereby loss of physiological integrity, basically viability, lead to impaired function and increased vulnerability to death. In more simple terms, it's basically when you take a tissue and you measure its ability to repair damage and to maintain its functionality, now it's outcompeted by the increased amount of damage, which really impair the functionality of the tissue and therefore contribute to this increased risk of death over the lifetime. And that leads over time to this whole range of problems that are listed here: decreased cognitive function, cardiac dysfunction. In fact, cardiac problem is one of the big killers of aging, with a lot of cardiac arrest around midlife, really contributing to a lot of early days. Medical progress have led to a lot of suppression of cardiac arrest, and therefore a lot more emergence of this more later effect: immune deficiency, blood disease, bone fragility, increased rate of cancer, muscle atrophy. Another big problem with aging is frailty, and therefore the problem in motility and activity in general. And there are more external signs of aging, like skin wrinkling, and this overall change in the appearance of people with old age. So my interest is about stem cells, which are really at the heart of maintenance of this tissue repair and are affected over the lifetime by the aging process. And the aging systems that you have been hearing from me over the last 3 lectures is the blood system, highly organized tissue, which stem cell at the top, which differentiate along many different paths to produce all the diversity of blood type you have in the organism. And here, what you are seeing in this movie is, again, this one stem cell giving rise to a lot of myeloid cell, granulocyte in particular in this condition. They are moving around, accelerating their cell division with increased commitment. They are living in special specialized microenvironment in the bone marrow cavity, which is modeled here by this trauma layer. And there is a lot of transcriptional events which really drive and are associated with this commitment to various lineage. So this is a system at steady state or geared toward producing myeloid cells, the content on my previous lecture. But today what I want to talk about is really what's happened with old age. And there is a lot of parallels between what we can find in a young mice and a young human being, and in an old mice and a whole human being. So age range is different. Mice age over a timeline of 30 months, like 2.5, max 3 years of age, while human age around a timing of like 100 years. So the scale is not identical at all, but the principles are very similar. What we see in both species is very much this accumulation, and by accumulation you see it by flow cytometry, looking within the marrow of a mice, a compartment that we know contains stem cell in a young mice, small compartment, much bigger compartment in an old mice, and here is a quantification of this increase over the 24 months. 24 months is already a fairly geriatric animal, where we have until midlife, 12 months, relatively similar number of stem cells which are in the bone marrow niche, but past midlife, we see this exponential increase in number. And these stem cells, which accumulate in large number, are defective. They are very poorly engrafting, leading to impaired regeneration potential of the system, and they are myeloid-biased, meaning they produce myeloid cells, more myeloid cells than normally, but as well much less lymphoid cell, therefore leading to a decline in immunocompetence and all the problems associated with old age and problem vaccinating the elderly, lack of immune response, because they produce much more and less capable lymphocyte. We as well see greater propensity to anemia, defective production of red blood cell, increased production of myeloid cell, as I mentioned, which as well fuel this higher predisposition to a whole range of myeloid disease, which are all myeloid disease of old age, myeloproliferative neoplasm, myelodysplasic syndrome, bone marrow failure syndrome, acute myeloid leukemia. And what has emerged more recently is very much aging of stem cell as a driver of age-related clonal hematopoiesis, where you have a clone of stem cell which expand, carrying a pre-leukemic mutation, usually affecting an epigenetic regulator, which are more competitive than normal unmutated stem cell, and are present with pretty much everyone over 70 years of age, and is very much associated with poor prognosis and a whole range of age-related disease, such as cardiovascular disease, gout, Alzheimer's disease, and so forth. So really important question to understand how stem cell age in order to try to combat all these features of blood aging. So there is a lot of work in the field. We are far to be the only lab working on this question, and a lot of cell intrinsic deregulation have been found, which contribute to this dysregulated activity of the hematopoietic stem cell, replication stress, DNA damage, impaired autophagy, metabolic reprogramming, and so forth. Conversely, there is a lot of problem with the milieu, the bone marrow niche into which this old hematopoietic stem cell reside, with as well bone marrow niche remodeling, chronic inflammation, loss of innervation, and so forth. So instead of listing them one by one, what I want to do is really like conceptualize how we are, in my lab as well, more generally in the field, starting to think about aging as a degradation of this stem cell niche regulatory unit, which is so essential to control and maintain the activity of the system, which trigger a set of adaptive response which have both beneficial and deleterious consequence. So you cannot have one without the other one. That's pretty much the take-home message here. And the example I want to show you is what we have done, demonstrating that it starts with a damaged bone marrow niche microenvironment, increased inflammation in the milieu, driving increased proliferation in the whole stem cell, leading to replication stress as one of the root cause for regenerative disease, regenerative defect with old age, but as well triggering cytoprotective autophagy engagement for metabolic adaptation and maintenance of stem cell quiescence. So what I mean by that? The bone marrow niche can be studied with many different approaches. Here is an example of single-cell RNA-seq analysis of a young bone marrow niche with all the different component part of the niche. And this is what remains in a 24-month-old animal, which is very few population in this part of the graph, which represent the vascular system as well as the endosteal, everything along the bone surface, and a massive expansion of this population in the central marrow, which is in large part why we have more stem cell accumulating in an old niche, because there is more of this mesenchymal stroma cell, which serve as a control mechanism for stem cell number. What we found as well is all of these cells in this system now secrete way more inflammatory factor, in particular interleukin-1, TNF, and so forth. So in response, as a crosstalk mechanism, in response to this niche inflammation, there is activation of the old hematopoietic stem cell to activate process of emergency myelopoiesis, which I have already detailed to you in my previous lecture, which reprogram this hierarchy of stem and progenitor and basically suppress the production of other blood cell type. You have less red blood cell, less lymphoid cell, and you amplify myeloid cell production, which is shown in part here with this increased GMP cluster in the bone marrow of an old mice. So basically, what we have as a consequence of an inflamed and remodeled old bone marrow niche is chronic activation of emergency myelopoiesis from an old stem cell, which is in large part responsible for this change in lineage output associated with aging. When we say activation of the stem cell, so they leave their quiescence phase, they enter S phase, they replicate their DNA, and there they hit another problem associated with aging, which is a less expression of MCM gene, which stands for mini chromosome maintenance gene, which are normally duper overexpressed in cells. They are rarely in rate-limited condition because they are this component of the replication origin complex, which binds the DNA and unwinds the DNA to allow progression of the replication forks. So what we found in all stem cell is they have much less of this MCM protein, therefore less origin of replication which are active at any given time, and much more of this feature of fork stalling and forks collapse, leading to DNA damage response, activation of the ATR pathway, and phosphorylation of phosphoxyc1, a lot of gamma H2AX deposited, indicating DNA damage, engagement of a lot of repair mechanisms, which are overwhelmed if you continue expanding the stem cell, if you put them in this liquid culture, as I show you in the movie, where the cell divide, accelerate, and differentiate. So what you see now from the stem cell population and their progeny is a lot of this forks collapse and stalling, leading to cell death. And we think that in large part why when we transplant all stem cell in a young recipient, we basically see much reduced engraftability from the old stem cell and as this regenerative defect. There is a second part of the picture here, which I will go very quickly because I really will delve into this result in the second half of my talk, which is when the old stem cell in vivo continue and return to quiescent, which is their default steady state position in the cell cycle, they carry with them a lot of gamma H2AX when they finish replicating their DNA. And this gamma H2AX is particularly locating on the rDNA gene. And then when they get back to quiescence, because the phosphatase, which normally dephosphorylate gamma H2AX, is mislocated as well in this old stem cell, I'm telling you, a lot of problem with a lot of cellular activity in this old hematopoietic stem cell. So the phosphatase is not efficiently dephosphorylating gamma H2AX, and as a consequence, quiescent HSC, old HSC, have this nucleolar gamma H2AX, which are persisting during like all their quiescent phase. So, and that's one of the metrics we use to really assess aging in the HSC system, persistence of this gamma H2AX, nucleolar gamma H2AX. So, this is what happens when you trigger proliferation, replication stress, problem with survival following a replication challenge, like transplantation. What inflammation in the niche as well does, it's trigger an adaptive protective response, which is engagement of autophagy. And the mechanism that we work out is very much because in response to inflammation within the old stem cell, you have activation of SOC3, dampening of the PI3-AKT pathway, change in FOXO3 nuclear localization, and in the certain cell, engagement of autophagy. In most cells, no engagement of autophagy. And when you have reduced AKT, you have reduced export of GLUT1, which is a transporter of glucose, and therefore leading to decreased glucose uptake, decreased glycolysis, and induction of oxidative phosphorylation-driven metabolism, and therefore everything associated with a higher activation and dysfunction in the old stem cell compartment, except for the few one, like roughly 30% of the population, which are able to engage autophagy, basically the one which sends a higher level of a niche inflammation. They engage autophagy and they have a much more controlled metabolism, and actually they are able to switch to beta oxidation and to maintain their functional quiescence using beta oxidation and lipid metabolism as a poor man capability of maintaining quiescence. So this is what I mean with this adaptive response, both beneficial and damaging. And with this kind of knowledge about the system, we can start figuring out effective anti-aging rejuvenation strategy. What can we do to make the system work better? And the three examples I want to tell you are blood-borne intervention, which are all the rage in the aging field at the moment. The parabiosis, the plasma transfer, the exercise, the caloric restriction. If you're interested by the aging literature, you see that over and over as a way to age less, rejuvenate your own organs, and so forth. Doesn't work at all for the blood system, but I'm getting to that. What we did in targeting niche inflammation or metabolic resetting to improve the regenerative potential of the system. So this is what we did a few years back when we started this work when I was still at UCSF, so quite a few years back now. And use this technique of parabiosis that I introduced in one of my previous lectures, which is the surgical ligation of the skin of two animals, just the skin, to establish a shared circulation between the two animals, which are basically joined together. And so in the context of aging research, we do this heterochronic parabiosis, where you tie together a young mice and an old mice. And that allows circulation of pro-use factor coming from a young mice into an old mice, or pro-aging factor coming from an old mice into young mice. And that has been shown to be able to rejuvenate, having a pro-use factor for a young mice, rejuvenate old neurostem cell, to rejuvenate some heart function, to rejuvenate some muscle repair function. So, and the aging factor coming from an old mice has been shown as well to trigger more neuronal inflammation in a young, in the brain of a young mice. So yes, there is systemic factor circulating which are impacting the functionality of young and old organs. So we did this experiment, and the killer experiment in the blood field, in the stem cell field, is very much stem cell engraftment. And you see the result here. All of the black and gray are basically young stem cell. They engraft very well. All the red are old stem cell. They engraft very poorly, very typical of the behaving of an old stem cell. And in orange is basically the treated condition. Gray and orange are these heterochronic parabiosis mice, where the gray is a young heterochronic stem cell, and the orange are all heterochronic stem cell. Absolutely no difference compared to all the old. So no effect of a systemic factor in an heterochronic context, no effect of injecting plasma taken from a young mice into an old mice to see if that restores the engraftability, still very bad. No effect of exercise in this condition, 7 weeks of free access to a running wheel. The mice look way healthier, younger looking, more shiny fur, like fit old mice. Their stem cells still very crappy, no rejuvenation at all. And similar for caloric restriction, which was a long, almost lifelong caloric restriction, absolutely no change between treated or untreated animal. So none of that systemic blood-borne rejuvenation intervention work, mostly because it doesn't reach the bone marrow cavity. And there is absolutely no change in bone marrow inflammation. What you really need to this approach with a parabiosis allows us to do as well is to look at crossover. So, which is the blood from the white mice going to the black mice, and vice versa. Same thing in this heterochronic context. The blood from an old red mice going into a young black mice. When you look at the crossover in the blood, it's roughly 40–50%. So pretty much mixing of the blood of the two animals. When you start to look at the crossover in the marrow, it's much lower, but it's around 10–15%, and it's consistent between an isochronic young pair or an heterochronic young-old pair. Where it's very different is the crossover in the stem cell compartment. Now, doing flow cytometry, looking at the stem cell, what we see is that a lot of the old stem cell spill over into a young bone marrow. This is roughly 30%. We can raise the number to almost 80% by doing a mobilization treatment with GCSF. And what does this old stem cell in a young mice do? They are represented here in blue. You almost do not see them, but they basically replace the young stem cell. We do not see a change in the size control, which is very constrained in young mice. There is a certain top number of stem cells that can live in this young bone, mostly dictated by the number of mesenchymal stem cell expressing leptin receptor. And this number is much higher in an old mice, but that what we see with this crossover of old stem cell is a replacement, and it's very specific to the stem cell compartment, because as soon as we start to look at downstream progenitor, we do not see anymore this large contribution of the old stem cell. So basically what we have is a lodging of an old stem cell in a young mice, which doesn't require irradiation of the niche or any of the preconditioning situation. And that allows allow us to study whether exposure to a young niche could actually improve the functionality of the old stem cell. So these are these old stem cell in a young niche in blue, which do expand a little bit, but when we test for their engraftment, there is absolutely no rescue. They are still old cells, they are still myeloid-biased in their output, and when we look at other metrics of aging, like this gamma H2X nucleolar foci I told you. The all heterochronic stem cells, which are large in the young niche, still have them. They still have delay cell cycle division. And when we do molecular analysis by RNA-seq, they basically still cluster with old stem cell. Although when we compare old stem cell in a young mice with an old stem cell staying in an old mice, basically they are very different. And what these old stem cells, which have lodged in the young mice, have is activation of every stress response pathways that you can imagine, which for sure made them very much more competitive compared to a young stem cell, which is basically nicely resting quiescent, no activated particular, and is being dislodged by this entering an hyper-competitive old stem cell. So, since this molecular analysis indicated that this old cell in the young mice were activated, we saw that maybe it's because they were early immigrant. And we know that when we transplant stem cells and they mount their regenerative response, they are early immigrant in the recipient bone marrow. If you try to re-isolate them and retransplant them, you cannot. When they are activated, they do not engraft. So we thought maybe they didn't have enough time to really take advantage of a nice remaining young bone marrow microenvironment. So we waited for 4 months after separation of the young and the old parabion. We kept this young parabion. We waited for 4 months and we analyzed these old stem cells which are present. So what we saw very clearly is a complete collapse of the contribution to peripheral blood. So the old stem cells are still there. They are in the marrow of a young mice 4 months later, but their contribution to the blood is pretty much negligible. They are kind of expanding. Also, we didn't have enough animal to really make these comments, but I think the key result is shown here. They still engraft extremely poorly, meaning functionally, even after 4 months of being bathed by all this good stuff in the young bone marrow, they still are all dysfunctional cells, like not being recovered in their functionality. So that's pretty much what this study told us. These old stem cells cannot be changed by just rejuvenating, by just being exposed to a young bone marrow niche microenvironment. Although what we learn when we study the aging niche microenvironment, where I show you the result at the beginning of this section, where we see degradation at the endosteum, all these cells become more inflammatory, interleukin-1 is being produced, interleukin-1 trigger emergency myelopoiesis activation from this old stem cell. If we block interleukin-1 signaling coming from the microenvironment, we actually can really restore normalize, I will say, blood production and improve the regenerative potential of this old stem cell. So this is shown here. We provide daily anakinra treatment. Anakinra is an inhibitor of interleukin-1 signaling, clinically used. So we block interleukin-1 signaling. When we do nothing, it does change nothing. When we do nothing else, it changes nothing. What we needed to do is to give a strong regenerative challenge. So we give anakinra, we challenge with 5-FU, and all the analyses are done 12 days later. When you take a young mice, challenge with 5-FU, 12 days later, you see basically that they have nice reconstitution, the regeneration is pretty good. If you block interleukin-1 signaling in this condition, there is no basic change. When you give 5-FU to an old mice, it's terrible. They really respond very poorly. You have almost no cell regeneration. Actually, you lose a lot of mice following old mice following 5-FU treatment. They mainly die from thrombosis because there is so much more platelet being produced. The blood of this animal becomes syrup, but their marrow is very nasty. If you block interleukin-1 signaling with anakinra, you can see a much more robust cellular regeneration in the marrow. And this is mostly increased production of red blood cell, erythroid progenitor, and lymphoid cells. So the blocking IL-1 signaling really rebalance the output of the system toward much more normalized blood production. And if you remove the ability of the stem cell to sense interleukin-1 signaling since the get-go, since the birth of the mice, using a straight knockout for interleukin-1 receptor, you actually see that with age, the stem cell is much less compromised in this functional ability. So the inflammation from the niche that you see over time really contribute to the functional damage which impacts the whole stem cell. But then, if you put this whole stem cell in a young niche with normal microenvironment regulation, it doesn't restore their functionality. What does restore the functionality of the whole stem cell is now to play with what we learn about autophagic regulation and the problem of the whole cell to import glucose and maintain glycolysis, which is really the fundamental mechanism of quiescence maintenance. So here is our experiment with fasting refeeding, where when you fast, you induce autophagy. And that's measured here in this reporter animal, this GFP LC3 reporter line, where the double membrane, autophagic double membrane, which will engulf cellular component, mitochondria, or other organelle, or macroprotein. So this double membrane has this LC3 molecule, lipid moiety, on the surface. It's tagged with a GFP fluorescence. So when autophagy is engaged, like with fasting, you see a downshift of GFP signal intensity. And when you fast and refeed, you see this upshift. So this is engagement of autophagy. Here is repression of autophagy, quantified here. Steady state, ad libitum feeding, fasting, engagement of autophagy, fasting refeeding, suppression of autophagy. That's the response in a young mice. When you do the same in an old mice, first they start with already engaged autophagy in roughly 30% of the population. You fast, they still engage further autophagy more globally to every cell. And when you fast refeed, you repress autophagy, but what is striking is the level of repression now bring it back in the vicinity of what you see in ad libitum fed young mice. And the result was very clear-cut when we transplanted this cell at the end of this fasting refeeding period, which is old ad libitum, poor engraftment. Old fasting, sorry for the blurriness here, old fasting, very poor engraftment. Old fasting refeeding, restore engraftment. Very similar to a young cell, which is basically not changed by fasting or fasting refeeding. So this tells us that, like, playing with this metabolic way of changing cell activity, what we see is basically we erase this problem with uptaking of glucose. The PS3, the PI3 kinase pathway is basically reawakened. We see more GLUT1 on the surface, more glucose uptake, restoration of glycolysis, and restoration of regenerative potential. It's unlikely that this will be persistent because this is just one round of fasting refeeding. So it remains to be demonstrated how much of this effect can be sustained, how much more we can do. But it's clear that fasting refeeding has entered clinical treatment for cancer patients because it allows to restore some much more active immune function in the context of chemotherapeutic treatment. So we think there is a lot more to understand about this metabolic resetting as a way to understand how to manipulate aging and how to improve the functionality of this old stem cell. So what we are doing now, we really are trying to, so first of all, we want to gain a broad understanding of both this intrinsic and extrinsic age-related deregulation and their cross-interaction, because that's the only way that we can understand what causes what and how we can intervene with them. So, and one of the approach which I'm not going to talk to you about because we are just finishing the heavy lifting with that, is we know a lot in separation of all of these processes. We know a lot in a young mice and in a very old mice. And there is a lot of assumption that the change will be linear, either increase, decrease, but that there is a correlation between what we see in young and what we see in old, as well as what happened in female and male. In mice, we tend to mix the sex, but that's not good habits to have. And so we have started this massive profiling along the life range from 2 months to 30 months of age, so very geriatric mice, every 2-3 months harvesting sample to try to deconvolute what happened over this age range and to identify potential target and inflection point to try to see, can we have a more like reason and approach of this problem of like, what are we trying to do? Are we trying to delay aging? Are we trying to restore functionality of an already old cell? Which kind of intervention, which kind of targeting approach can be used? So stay tuned. We are wrapping up this kind of study. What we have done as well is try to identify additional age-related resilience mechanisms to harness in order to like make an old cell work better. And what I want to finish my presentation to tell you about is a new story about the cytoprotective nucleolar stress response which maintains stemness during hematopoietic stem cell aging. And it's going back to what I showed you previously about this cycle of activation in the old stem cell compartment, and in particular the fact that this gamma H2AX is carried over on their DNA gene and become repackaged because improperly dephosphorylated in the nucleoli. So when it's there, we have no evidence that there is persisting DNA damage. We have every evidence to think that what gamma H2AX does is an histone mark which signals the transcription machinery to stop transcribe, because there was a DNA damage problem. Basically, the role of gamma H2AX mark is to basically serve as a landing platform of all the DNA repair mechanisms. It come in as soon as there is a DNA damage, serve as a landing platform for the DNA repair machinery, and as well signal to the transcription machinery, do not transcribe, we are repairing here, we do not want a collapse of the transcription and the repair machinery. So we think that the repair is done. The cell go back, but what since the mark is left not fully dephosphorylated, it's basically act as a transcription, like as an epigenetic mark telling the transcription machinery, do not transcribe. And we have evidence with a decreased transcription of the rRNA gene, which is observed in old hematopoietic stem cell compared to young stem cell. So the nucleoli is one of the biggest structure in the nucleus, and it's a site of ribosome biogenesis, extremely important for proteostasis and controlling the rate of protein synthesis and serving as a big hub of stress response to integrate change in nutrient, like a lot of cellular stress. What we know essentially from the work of Sean Morrison is that stem cell, HSC, require very stringent control of protein translation to work at peak efficiency. Is this Gaussian distribution? Too little protein translation, not good. Too much protein translation, not good. Right in the middle, and a strong control of how much protein is being made. So we were wondering whether this persisting gamma H2AX at the nucleoli were changing the rate of protein translation in old stem cell. So what we did was to sort the stem cell from young and old mice. Again, when I say young, it's a two months old mice, and old is a 24 months mice. So geriatric, old. We sorted the stem cell, put them in culture with OP-Puro, which allow us to measure nascent protein synthesis. So you see young stem cell in this culture condition, a lot of protein synthesis, a lot of this green signal. In old stem cell, when you don't have the nucleolar gamma H2AX foci, here it's just a staining for gamma H2AX, but trust me, it's at the nucleoli for most of these, for the big dot. When there is no gamma H2AX, there is translation, less than in young, but protein translation. But when there is this gamma H2AX nucleolar signal, there is much reduced protein translation. And we can confirm that by using a flow cytometry method compared to imaging approach for OP-Puro in culture, or in vivo by injecting OP-Puro and sorting the stem cell. In both cases, the results are the same. In the stem cell compartment, we see decreased protein translation in all stem cell. We see absolutely no change in the downstream progenitor. Again, highlighting the fact that these defects are very specific to the stem cell compartment and rarely shared by the downstream progenitor. Not to say that the downstream progenitor are totally fine, but they have other type of issue that what we are mapping in the stem cell compartment. We as well confirm that this decreased protein translation in stem cell is observed as well in human. Here we don't have as much sample, and our old is not as old as our geriatric 24 months old mice. It's a 66-year-old bone marrow donor. And when we do the same type of OP-Puro incorporation in vitro, we as well see that the all stem cell have, all human stem cell have reduced protein translation. So the next question was, what's the mechanism supporting this reduced protein translation? So there is a lot of pathway which funnel into a control of protein translation. I told you is one of the most regulated cellular activity because that's life and death for a cell. So you have TOR signaling, which is a key control of tuning the rate of protein translation. Actually, in all stem cell, what we see is increased TOR activity, unlikely, therefore, to be the one which will be reducing protein translation because you will expect decreased protein translation. We as well see no involvement of the integrated stress response. We don't see unfolded protein or protein aggregates in this whole stem cell, which therefore leaves us with this connection which initially felt fairly puzzling between this nucleolar gamma H2AX and this reduced transcription of the rDNA gene. So, textbook reading, my postdoc did a lot, and PhD student did a lot of work figuring out, and that leads us to this nucleolar stress response, which is a very ancient mechanism as well to basically link the transcription of the rRNA gene with the availability of the ribosome binding protein, essentially RPL5, RPL11. It's basically a sensing mechanism to see if the rate of protein translation is matching the amount of RNA being transcribed in the nucleoli. So when everything is fine, these proteins are bound on the rDNA, and you have no stop in the cellular activity. When you have an imbalance in transcribing rRNA gene, then the RPL5 and 11 protein basically are free now to bind MDM2. And binding MDM2 free a p53 response. They repress p53 activity, therefore engaging this nucleolar stress response. So this process is in two stages. You first have nucleolar stress, which is this inhibition of rRNA production associated with reduction in protein synthesis and dispersion of a lot of the nucleolar component, which is associated with this change in basically rRNA production. and then the engagement of the downstream p53-like response, which is this nucleolar stress response. Okay, we started looking at that, and what we found in this old hematopoietic stem cell is every feature of nucleolar stress. We have decreased rRNA content. This is a tape station method I already showed you. We now have qRT-PCR as well as a new method to look back by flow cytometry for measuring rRNA production, decrease in all stem cell. We look at the RPL5 and RPL11 synthesis, and this is basically increase in all stem cells, so less rRNA, more ribosomal binding protein. We have this massive nucleolar dispersion of nucleolar component. Here I show you with NPM1, but we have the same with fibrillary, so the size of the nucleoli is much bigger in this old stem cell, which is kind of quantified here. We have more, we have additional quantification method. And when we look at published RNA-seq dataset comparing young and old, ours, but a lot of other in the literature, there is blazing activation of the p53 response in this old stem cell. So all of that suggests that indeed there is constitutive nucleolar stress in old hematopoietic stem cell. So we tested that. What does induction of nucleolar stress do to a stem cell? And we tested that by taking young stem cell, put them in culture with actinomycin D, low doses of actinomycin D in the range of 1 nanomolar to 5 nanomolar, basically block polymerase 1, POL1, and basically decrease translation of the rRNA gene. We have as well tested 2 other POL1 inhibitors with very, very similar results. So it's not just actinomycin D. And so when you expose young stem cell to actinomycin D, what you see is progressively this massive dispersion of the nucleolar component, indicating nucleolar stress associated with reduced S-phase entry, similar to what you constitutively have in an old stem cell, reduced protein translation, and reduced expansion of the cell in methylcellulose. Basically telling us that either through aging with our nucleolar gamma H2X feature or by inhibiting POL1 with low doses of actinomycin D or other POL1 inhibitor, we have this feature of nucleolar stress, impaired proliferation, decreased translation, and defective expansion. The problem of drug treatment is as soon as you remove it from the drug, the young stem cell revert back to their normal function, so we couldn't test the consequence of inducing replication stress on their functionality via transplantation. They basically engraft totally normal and don't maintain this feature. So to test the role of this nucleolar stress response, we turn to a different genetic model, which is this mutant mice of MDM2 mutant mice, which has this C305F mutation in the binding site of the ribosomal binding protein. Therefore, when there is this imbalance triggering the nucleolar stress response, the MDM2 cannot bind the RPL protein and therefore cannot engage the nucleolar stress response. And I don't want to bore you, but we did all the control. Basically, these mutant MDM2 mice are insensitive to induction of nucleolar stress, but remains very sensitive to induction of other type of p53-dependent stress, such as irradiation. They have very similar response and engagement of a p53 response. So what happened to the mice? Nothing. Like we look by flow cytometry, CBC, with their blood parameter, their hematopoietic system is damn normal, which is always very depressing for the student doing the work. It's like, did I waste all my time getting these mice? No, he didn't. Because when he started transplanting, now they can see that there is a lot of problem with engraftability of this p53, this MDM2 mutant stem cell. In primary recipient, much decrease, and in secondary recipient, following either transplantation of whole bone marrow or a re-isolate donor stem cell. So basically, even in young mice, the stem cell needs the ability to engage this nucleolar stress response for peak regenerative capability that you assess by transplantation. What happened in an old mice? The number of stem cells in an old mice, which is now shown in purple, is much, much more increased. And this we still don't have an explanation why we accumulate so much more old stem cell in this MDM2 mutant mice. But certainly, humongous number of stem cells, they are still, most of them very quiescent, and they still have all the feature of nucleolar stress, with the gamma H2X at the nucleoli as an indication of like the underlying stress is still there. There's problem with gamma H2X limiting the production of rDNA, triggering nucleolar stress is still there. What they do not have is their ability to respond to this stress by mounting this p53-mediated response. And because they do not have this ability, when we transplant this old mutant stem cell, they completely crash. There is absolutely nothing remaining in terms of functionality. So you have a mouse which accumulates humongously dysfunctional old stem cell, but is still pretty normal and live exactly the lifetime, but cannot mount a regenerative response. We have started treating them with 5-FU and other challenge, and there it's not good. I mean, compared to a normal wild-type old mice. So what this tells us is very much that nucleolar stress has some damaging effect on stem cell biology, which is impair protein translation, impair ribosome biogenesis, and decrease protein translation. But the engagement of this MDM2 p53 mediated response, it's absolutely essential to retain stemness and the ability to regenerate the blood system and to control as well through mechanisms that we still don't understand HSC number. So that was based on transplantation. We were very interested to try to assess this retention in stemness in a non-transplantation context, when you really increase proliferation. And inducing activation and proliferation in an old mice is not as easy as doing that in a young mice. Basically, I already alluded to that, when you give 5-FU, which is one of the very clear way to induce activation and proliferation, most of the old mice die, and it's thrombosis really which kills these mice. Poly-IC, which has been shown, this big massive interferon-mediated activation, is absolutely lethal to an old mice. We give poly-IC, the mice drop dead in 2 days. So what we reverted to is this old-fashioned cyclophosphamide GCSF, which is very old mobilization protocol. You give a cytotoxic agent, and then you follow by daily injection of GCSF. And this is a very stereotypic activation response where at 2 days, basically 2 injections of GCSF after 1 injection of cyclophosphamide, all the stem cells in the marrow enter cell cycle and activate. And by day 4, meaning 2 more injections of GCSF, they basically return to quiescence in the marrow, and there is a wave of stem cells leaving the marrow, which are present in the periphery, and that's what has been used clinically to harvest stem cell mobilized in the periphery before the onset of other mobilization treatment. So we use this approach, and we did a lot of single-cell RNA-seq to try to see if we can pick on these biologies that we were interested. So this is relatively complicated, so I will try to be to explain it simply. So this is a separation day 2, day 0, day 2, day 4. At day 0, young are in black, old are in red. We see that stem cells are very different in their maintenance of quiescence. That's not new, has been shown over and over. The young are glycolytic, the old are this weird metabolic state that we can use for metabolic resetting. So that transcriptionally appear very, very clearly. At day 4, when they are returning to quiescence, we start to see again this separation between young and old stem cell. But at day 2, when they activate, there is basically this intermingling of young and old cell in terms of distribution in the UMAP. Looking with Leuven cluster in this context, what we found at this day 2 is this little cluster called cluster 9, which is only observed, this is blue cluster, which is only observed in old stem cell. And this cell, this cluster 9 cell, are basically almost entirely quiescent. So in this day 2 activation protocol, what we identify is an activation resistant old hematopoietic stem cell. So that's their molecular signature, and it led credence to what we did after, which is to use the Fuji mice, the Fuji2 reporter mice, which is a cell cycle reporter mouse model, which allows to follow progression from G0, SG2M, and G1 in the compartment, to use them to try to identify this activation-resistant old stem cell. So young stem cell, young HSC in an unchallenged mice, most of them are in quiescence. You have a little bit in G1 and is SG2M. You do the day 2 mobilization treatment, everything activates. So you have no quiescent left in the young, and basically the cells are either in SG2M or in G1 phase. All mice, all stem cell, mostly in quiescent, very few again in G1, SG2M. You activate day 2 treatment, we see this remaining small percent of cells which are still in quiescent and which we do not have at all in the young. It's a few percent. Remember, this is a vastly extended compartment. When we quantify the number of cells corresponding to this few percent of activation-resistant old stem cell, it's actually very similar in number to this quiescent young stem cell. So it's not a negligible number of cells. And using this Fuji reporter mice allow us to really isolate this cell. And the first thing we did was molecular profiling with a SMART-seq analysis of this isolated quiescent G0 old HSC after this day 2 treatment. They are not as pure as our cluster 9 cell, but a large fraction, more than 50%, are quiescent. The rest is in S phase. And basically, the quiescent part of this activation-resistant isolate cell colocalize very much with the cluster 9 cell. So we have a way to isolate this activation-resistant old stem cell, which now we can test functionally through transplantation. So comparing in both young and old at day 0 or day 2, either bulk, which means everything here, or J0, which means what's in this green gate. So in the young context, at day 0, separating quiescent from the bulk, no difference in engraftment, it's the same. At day 2, we can only get the bulk, and as expected, activated young stem cells do not engraft well. It's very poor engraftment. In the old, same thing, separating bulk or G0 at day, without any challenge, no difference, engraftment is terrible. But at day 2, when we compare bulk, where there is really nothing left, to this activation-resistant J0 subset, we have everything left with what is engraftable in an old mice. So these are duper hyper-competent old stem cells, which are carrying all the remaining regenerative potentiality of the system. They are all the features of nuclear stress. Here I'm only showing you the dysmorphic NP1 staining which is this diffusion of the nucleolar component, but as well we have the gamma H2A staining here. They have this very low protein translation compared to an activated whole stem cell, which are increasing protein translation. And we derive from our single cell analysis a signature of nucleolar stress response, which we are now using in many different contexts, but basically with this signature, they are blazing in terms of activation of this very specific p53-dependent nucleolar stress response score. When we remove the ability to mount this nucleolar stress response, basically using this MDM2 mutant mice, and do the same day 2 mobilization treatment and look at what's happening to this population of activation-resistant stem cell. Unfortunately, we didn't cross these mutant mice with the Fuji mice. That would have been needed, but since this cross will take 2 years before we can get the mice, we went with this molecular analysis, and basically molecularly what we can see is that we are losing this activation resistance subset, and we are restoring basically response to death when you challenge the system, as well as increased proliferation, increased protein translation. So basically we are losing this protective mechanism. So that's the take-home message of this work, which is this nucleolar gamma H2AX really create nucleolar stress. And nucleolar stress lead to this impaired ribosome biogenesis, problem in protein translation, and trigger this nucleolar stress response. That I didn't show you, it's in the same subset which as well engage this autophagy response. So here we are seeing converging mechanisms of cellular maintenance, which are reactive to a changed bone marrow niche microenvironment and really allow the cell, this whole stem cell, to still maintain themselves in this very damaged niche environment and maintain themselves through convergence of reduced protein translation, increased threshold of cell cycle activation, basically allowing a subset of all stem cells to not be constantly activated by the inflammation of the niche and to be preserved and to preserve their engraftability and regeneration potential. So if we can harvest this kind of resilience mechanism to enhance them in more of the stem cell compartment, we may be able to restore a lot more regenerative potential in an old individual. And that's really where we are going with this work, showing the correlation with human biology and trying to find the way to actually raise cell cycle activation in more of the compartment through this kind of mechanisms that we have identified. So to finish, we are continuing to decorate this kind of like crosstalk mechanism between the damaging feature and the cytoprotective feature. It's complex biology, but it's very much interconnected. And if we ever want to have translational goals, which are basically health span, we really need to understand that. And we need to move beyond just transplantation. It's a beautiful technology to test the functionality of the cell, and it's great clinical application for a lot of problems. But in the context of health span and maintaining the longevity of the blood system, and it's not going to be the solution. We cannot transplant. We need to really in situ activate and make the whole stem cell able to more produce balanced blood production, to have this enhanced regenerative mechanism which allows them to maintain immune defense as well as prevent clonal hematopoiesis and fight blood and prevent blood cancer development. So in the field, there is a lot of strategy which are being tested right, left. This is one of such a review with all the different type of approach which are being tested in many different organs. I think we are just at the beginning of a very long road to understand what approach will be working on stem cell in these various organs, because it's not clear at all that one bullet will hit all the problem in these different organs, especially because aging is asynchronous within tissue. Some organs age first, and then there is a cascading event. So really a lot more basic / translational research is needed. And I am over my time, so I'm stopping now. Want to thank Carl, Melissa, which really were contributing a lot to the last part of my talk. Thank you for your attention, and it has been my great pleasure to give this for lecture. And thanks Hugues and Denis for their fantastic invitation. It has been a treat to be here with you for the last 3 weeks. Thank you.